Hello, and welcome back. In this module, we're really going to take a deep dive into the innate immune system. So for the next 2 weeks, we're going to focus on the molecular mechanisms and all the cells and their effector functions that constitute our most basic and earliest responses to infection. In particular, this week, we're going to focus on barrier tissues and antimicrobial strategies that make up really the oldest, the most evolutionarily ancient features of our innate immune response. Though, as you'll see, being evolutionarily ancient doesn't make things simple. But nevertheless, we're going to dive right in. And what I really want to do is situate the innate immune response in the context of sort of several layers of immunity that we have. So the most basic protection that we have against pathogenic microbes are simply the anatomical barriers that make up the surfaces of our body. So our skin is really the largest immune organ that we have. It keeps things on the outside and prevents them from coming in. But the linings of our gastrointestinal tract, our respiratory tract, these are also coming into contact with pathogens on a really regular basis. And they serve as additional barriers that prevent microbes from coming into our internal body tissues. Sometimes these barriers fail, however, and when that happens, we rely on a set of really broadly specific antimicrobial molecules. What I mean by this is these are molecules that we secrete, that we make on the surface of our skin and other tissues that are good at killing large classes of microbes. They're almost like, the Purell of our immune system. They don't kill anything in particular, but they kill a lot of things in a really non-specific way. These include a set of proteins called complement, which we're going to focus on in the next couple of lectures, as well as a number of other classes of antimicrobial proteins, which we'll discuss a couple of examples of in this one. The idea here is that these are fairly simple mechanisms that are very important first lines of defense, and frequently they're enough to overcome an infection. However, when they're not enough, our next line of action is to recruit cells of the innate immune system. This includes phagocytes like macrophages, as well as all those granulocytes we learned about in the hematopoiesis lecture, as well as innate immune cells of the lymphoid lineage called natural killer cells, which, remember, are capable of recognizing cells which have downregulated MHC class I. These cells are able to respond to pathogens in a more specialized way, but still in a relatively non-specific way. And importantly, they do so in an antigen non-specific way. They're good at killing bacteria or viruses, cells infected with viruses, but they do so through fairly stereotyped mechanisms, and they do so early in the course of infection. Innate immune cells are very good at their jobs, but sometimes even they are not enough. And in those cases, we next need to recruit cells of our adaptive immune system. And this includes B cells, which make antibodies, as well as T cells. And the effector mechanisms of adaptive immunity usually, in almost all cases, take care of any infection that we might see. And that's the reason that most of us are alive and healthy. Although, as we'll see at the very end of the semester, a lot of human diseases actually arise from failures of one or more of these layers of immunity. So your book breaks down these steps in a way that I think is actually really helpful to think about the way this happens. And basically, this is a checkpoint system. So if we start with infection with a pathogen, normally we would contain that infection by exclusion by an anatomical barrier. If that barrier fails, if the barrier is successful, we have prevention of infection. But if that barrier fails, we have instead the establishment of infection. And if we have an established infection, the very first thing we're going to do are mount highly non-specific processes, which this figure calls broadly specific effectors. Again, things that are just really generally good at killing pathogens, killing microbes in a non-specific way, but that we have at the ready at a moment's notice to take care of a pathogen infection as soon as it starts. If those things are successful, we remove the infectious agent. If not, our next line of defense is actually to recruit the effector cells of the innate immune system. So recruitment of cellular immune responses takes a little bit of time. So you can see here, prior to this, we were talking about really instant reactions to infection. The recruitment of innate immune cells takes anywhere from a few hours to a few days following the establishment of infection. But these cells are really good at recognizing pathogens, recognizing non-self entities, as we've said. In particular, they recognize features of pathogens called PAMPs, or pathogen-associated molecular patterns. And when they do so, they become activated and start to exert their effector functions. So if they are phagocytes, they'll eat things. If they're granulocytes, they might release the proteins in their granules, which include a lot of cytotoxic effectors, which can kill pathogens or infected cells. Ultimately, these responses are pretty good at removing infectious agents, but if they don't work, we now need to invoke our adaptive immune response. And so that will involve the transport of antigen into the lymphoid organs by antigen-presenting cells. Those antigen-presenting cells will present the antigen to naive B and T cells, and those activated B and T cells will undergo clonal expansion and differentiation into their various effector cell subtypes. So maybe a plasma cell for a B cell, maybe a cytotoxic lymphocyte for a T cell. And as I've said, usually by the time we get to this stage, adaptive immune responses are quite potent and they're very targeted. And so almost any infection that we encounter can be taken care of by an adaptive immune response. So if we want to look at this kind of in more pictorial form, if you're not a fan of flowcharts, you can think of it this way. If we have our body tissue here, we'll imagine this the skin. We have a bacterial colonization on the surface of the skin by some sort of pathogenic bacterium that we don't want there. The best way to prevent infection is just to keep it on the outside. And so as long as the skin is intact, usually that's enough. But in some cases, either because we have a cut or a burn, or because the bacterium is actually actively trying to invade, we can have a breakdown of the barrier tissue and the establishment of an internal infection. And in that case, remember, our first line of defense are highly stereotyped, broadly specific effector mechanisms, things like the complement system, antimicrobial peptides, and any resident cells like macrophages or dendritic cells that happen to be in the tissue. They'll get started immediately. And oftentimes, as I've said, this is enough, but if it's not, we need to then start recruiting new immune cells into the tissue. And that process of recruiting new immune cells into the tissue is one of the things that we call inflammation, which we'll get into quite a bit later on. But inflammation involves the recruitment of new cells. We can sort of change the numbers game. We have new cells that have new effector functions, but are still acting as part of the innate response. They're not acting in an antigen-specific manner. They just see, hey, this is a bacteria. Hey, this is a virus. I'm just going to activate and do what I do. And in the meantime, some of those innate immune cells, like dendritic cells, can carry antigens from the infection to the secondary lymphoid organs, present them to a naive T or B cell, which then becomes activated and traffics back to the site of infection to exert its effector function. So a cytotoxic response, the secretion of antibody, what have you. So in this set of modules 2 and 3, we're going to focus on innate immunity, which is mostly these first 3 boxes. And this week, as I said, we're going to focus on kind of the earliest, most basic, most conserved features of our immune response, which are barrier tissues and basic, non-specific antimicrobial proteins and strategies. So these first 2 boxes, sort of. So in the next slide, what I want to show you is that our barrier surfaces, in addition to being barriers, have a number of chemical strategies that prevent colonization of our tissues by pathogenic microbes. And so there's a lot of them here. You can read them. No need to memorize all of them, but you can see that the chemical strategies that our tissues take are adapted to the different microenvironments that exist in different parts of our body. So in our stomach, of course, we have low pH. In our intestines, we have digestive enzymes. These are good at taking care of microbes. In our airway, we have mucus and ciliated membranes, which sort of sweep pathogens out, so on and so forth. But instead of memorizing all of these different strategies, what I want to do is highlight some general concepts that unify the ways that barrier tissues prevent the establishment of infection. So if we categorize them broadly this way, our skin, gut, lungs, as well as our sort of, facial cavities, all of them employ mechanical strategies to prevent colonization by pathogens. one thing that unites all of our barrier tissues is that, for the most part, they all consist of cells called epithelial cells. And epithelial cells are joined by cellular structures called tight junctions. Tight junctions exist between epithelial cells at their borders, and they prevent the diffusion of anything, liquid or anything like cells or pathogens between the paracellular borders of cells. And in this way, they basically form a wall. Nothing can get past the epithelial barrier, at least not without perturbing it somehow. So that's a mechanical force. In addition to just serving sort of as a fence, the mechanical forces of the flow of air and fluid through our body also tend to sort of flush or wash out pathogens from places where we don't want them to be. So that might be breathing, coughing, sneezing. It might be mucus moving out in all the ways that it does. Tears have antimicrobial function in one way, just by washing your eyes, just by washing anything in your eye out that might be there. These all count as mechanical strategies for limiting infection. However, these tissues all have chemical features, some that are generic and some that are specific. I mentioned low pH before. The lungs are coated with surfactants, which prevent things from sticking very effectively. Tears, in addition to just washing our eyes, have enzymes in them that break down microbes. And more broadly than that, our bodies are constantly secreting classes of molecules that have broad spectrum antimicrobial activity. Defensins are one example of this, so there are different types of defensins in different parts of the body. We're going to talk about a couple of examples of these antimicrobial proteins later in this lecture. But overall, what I want you to appreciate is that we have these chemical strategies in all of our various body tissues that are very effective as a very primitive first line of defense against microbes. So in addition to mechanical and chemical features, we also have microbiological strategies, and this includes the colonization of our skin by what we call our healthy bacteria, or our normal microbiome. So the bacteria that normally live on our skin and our other barrier tissues are really highly adapted to living in the ecological niche, which is our bodies, basically. And for that reason, they're very good at outcompeting pathogenic bacteria, new bacteria coming in that we really don't want. So they can outcompete them for resources, or they may exert active strategies to exclude colonization by new microbes. But whatever they're doing, the normal microbiota on our bodies are a very important part of the sort of intrinsic way that we prevent the establishment of infection, so that they should not go underappreciated. So all of these features can be seen in very simple microbiological experiments. This is one that I don't recommend that you do, but I do think it sort of demonstrates some of the things that we're talking about. So our skin, as I mentioned, is coated with all sorts of antimicrobial chemicals and enzymes, and they have broad activity against many types of microbes, but not all. You can sort of see this in action in this experiment, which is depicted here, where if you inoculate the tips of your fingers with either Staphylococcus aureus or E. coli, you let it sit there for 30 minutes, and then you touch a fresh agar plate, you'll see that the Staph aureus infection, it maintains itself on the fingers. Staph can live on our skin for a while, whereas E. coli dies pretty quickly, within 30 minutes. We have a lot of strategies that are very good at killing E. coli infections. Again, this is happening within 30 minutes. This does not involve inflammation. It does not involve any cellular response, and certainly not an adaptive response. These are things that are happening very, very quickly using very intrinsic programmed mechanisms. So, why E. coli and not Staph aureus is not really the topic of this lecture, but E. coli and bacteria like it are susceptible to a number of different stereotyped antimicrobial molecular mechanisms that we're going to talk about, with two examples of antimicrobial proteins, chemicals that our body secretes that make up the earliest parts of our innate immune response. So one that I want to highlight is called lysozyme. Lysozyme is an enzyme which is very good at degrading the cell wall of bacteria. So remember, unlike eukaryotic cells, bacteria have a plasma membrane like we do, but also a cell wall. And this cell wall is made up of structures composed of peptidoglycan. And peptidoglycan happens to be the substrate for an enzyme that we make called lysozyme. So lysozyme recognizes peptidoglycan in the cell wall of both gram-positive and gram-negative bacteria. When it does so, it chews it up and it exposes the plasma membrane of the bacterium. And that exposed plasma membrane is then susceptible to all sorts of mechanisms of bacterial killing that we have at our disposal, which would normally not be possible because the cell wall would prevent access to the plasma membrane of the bacterium. So lysozyme is a very effective strategy for making bacteria susceptible to lysis. One of those lysis mechanisms is a different class of antimicrobial chemicals that we make called defensins. So you saw that there were different types of defensins in different tissues, but a common mechanism of action involves the positive charge of the defensin molecule, which allows it to integrate into the plasma membrane. When it does so, it can sort of polymerize in ways that form pores within the membrane, sort of pinches off the lipid membrane here. And when we form a pore in a membrane, that disturbs the osmotic balance of the cell. Usually that causes water to rush in, the cell swells and pops. And so this mechanism of bacterial lysis is a very effective antimicrobial strategy that is mediated by molecules called defensins. So we've talked about physical strategies, a couple of examples of chemical strategies, and one additional point I want to make in this lecture is that the adaptive immune system can actually co-opt or sort of utilize the effector mechanisms of the innate immune system in ways that sort of tie the two systems back together. The line between innate and adaptive is not actually all that clear-cut. This is very obvious when we think about the ways that antibodies fight infection. So antibodies, remember, are antigen specific. They only bind to a single antigen. They're made by B cells, which are antigen specific. So they are definitely adaptive immune components. But the way they actually fight infection is by using innate immune effector mechanisms. So let's look at a few examples of this. One is that antibodies can bind directly to microbial products like bacterial toxins. Normally these toxins would bind to receptors on the surface of cells, which those receptors would then mediate their toxic functions. If we have antibodies which recognize these toxins, instead the antibodies bind them up in a multivalent fashion and trap them in these things called immune complexes. So these macromolecular structures of antigen and antibody all bound to each other are called immune complexes. And by binding them up in immune complexes, they are effectively neutralized. We call this process neutralization because now they're unable to bind to their target receptor on the cell surface. In contrast, antibodies can also bind directly to pathogens, pretty famously. And as it turns out, when we coat something with antibodies, antibodies just look really yummy to macrophages. And so we call this a cellular eat me signal. And when we coat something with a molecule that makes it really attractive for phagocytosis, that process is called opsonization. So something that's been opsonized has been marked for eating by a macrophage or another phagocyte. So antibodies are very good opsonization agents. The other way that antibodies make use of the innate immune system, particularly in the blood, is by initiating a cascade of proteins that are called complement. So complement constitute a major feature of the innate immune response. We're going to focus on them in a lot of detail in the next couple of lectures. But for now, suffice it to say that complement can kill bacteria using a lot of different effector mechanisms. And any bacterium coated by antibody is very strongly then targeted by the complement system in ways that ultimately lead to their demise and control of infection. An interesting way that all of these things are linked is that all of these actions of antibody neutralization, opsonization, and complement activation really end up activating macrophages in the process of phagocytosis. And so immune complexes are, again, really yummy to macrophages. They like to eat them and break down whatever they take in their lysosomes. Opsonized pathogens are eaten through phagocytosis and broken down. In complement, one of the effector mechanisms of complement is also opsonization, and also then targeting the things that the complement system has opsonized for eating by macrophages and being broken down that way. So a lot of times, you can see here that macrophages are the cleanup crew. No matter what strategy we take to kill a pathogen, ultimately we need our macrophages and other phagocytes to come in and eat and process the debris and then sort of the wreckage of that process. Okay, so let's summarize these basic principles that we've introduced in this lecture. Organisms are protected by multiple layers of immunity, as we saw. These include barriers and chemical defenses, which naturally are our very, very first line of defense. They just keep things at bay, keep things out of our bodies that we don't want there. When these fail, we have very ancient intrinsic innate immune mechanisms, as well as a variety of innate immune cells, which provide broad protection, broad antigen non-specific protection. If those components of the innate immune response fail, we then need to invoke our adaptive immune system. And remember that the adaptive immune system needs the innate immune system and primarily in order to present antigen so that we can activate naive lymphocytes of the adaptive immune system. So going back to the barrier tissues, the way they function primarily to prevent microbial colonization are through a combination of mechanical and chemical defenses, which either kill pathogens or simply remove them from our bodies somehow. And in addition to this, our barrier tissues are coated with native flora, our native microbiome, which outcompete potential pathogenic flora that are trying to colonize us. Antimicrobial agents that we secrete on our barrier surfaces and in our body fluids provide broad protection against any potential infection. Two examples of this that we discussed are lysozyme, which degrades bacterial cell walls, as well as molecules called defensins, which disturb bacterial membrane integrity. And finally, another basic principle are that antibodies are really able to co-opt all of these processes, and these are a way that the adaptive immune system uses the effector mechanisms of the innate immune system to promote an immune response. So two of the things that we focused on there were that antibodies were very good at opsonization, marking their targets for phagocytosis by innate immune cells like macrophages, as well as by inducing the process of complement fixation, which, as I said, induces multiple mechanisms of pathogen control. So in the next couple of lectures, we'll dig in what on earth is complement. And so complement is one of the the sort of watershed immunological processes. So anybody who takes an immunology class, it's sort of a rite of passage to learn about the complement system. There's a lot going on. We're going to go through it step by step, but it's actually really cool. So we'll talk about complement in the next lecture. Thanks.